Put the lights on. Put your lights. And we'll take these guys off. Oh, it's a bit dark in here, but that comes out of the lecture lights on. Okay, so I asked the TAs to get the exams back to you on Thursday. So hopefully, unless you hear from me otherwise, we'll do that. And I haven't had a look at them in any detail, so I don't know much about them yet. But I'll tell you all about it on Thursday and let you know where we go from there. So do come in and pick up your exam. Otherwise, I get stuck with a whole bunch that I then have to give away at odd moments. Today we're going to be talking about finishing up the story about audition, and then doing a little bit on olfaction, and then getting away from the senses, moving on to the motor system. So where we got to in terms of audition really was the movement of the basilar membrane, and the way the movement of the basilar membrane as sound comes down the cochlea here, the way that movement is converted into a standing wave, where the amplitude of the standing wave of vibration of the basilar membrane depends upon the frequency of the sound. So that high frequencies tend to make to create a larger amplitude of the standing wave of movement here, the sort of envelope of movement here, and less of an amplitude down here at the end. And that's shown in the next diagram, where the x-axis, the y-axis here shows the amplitude of this sort of envelope of movement to the basilar membrane, this so-called standing wave of movement. And the y-axis shows the distance from the x-axis shows the distance from the stapes, that is the distance from here on out. So this part here is that part there. And as you can see, the lower frequency, and the ear is being presented with a series of sine waves, single tones, of different frequencies. And so the lower frequency sounds tend to create a larger amplitude envelope of vibration at points of the basilar membrane that are distant from the stapes, and the higher frequency sound creates an envelope of vibration that has a higher amplitude closer to the beginning of the basilar membrane by the stapes there. And one way of thinking about this is to think that maybe the basilar membrane itself is slightly stiffer in this part of the cochlea than in this part of the cochlea. And you know how a stiff tuning fork will vibrate at high frequencies, and a less stiff tuning fork will tend to vibrate at lower frequencies. And so when sound comes in, movement of the membrane down here in the less stiff part resonates with the sound frequencies, which are lower, and the stiffer part here, the movement resonates best with sound frequencies that are higher. So that's often how this difference is explained. But, as I explained to you, this is a very complex system of fluid movement and membrane movement, and really that's sort of just a way of memorizing that's how it happens rather than an actual mechanical description. But that is the way it is. So, what about the hair cells? The hair cells are mechanically coupled to the tectorial membrane and the basilar membrane. And so, as we've seen, as the basilar membrane moves, so the hairs on the hair cells get displaced back and forth. And that creates a change in their membrane potential, which, in the depolarizing direction, will trigger action potentials in the hair cells. And so, what we're looking in this diagram here, these diagrams are called tuning curves, auditory tuning curves. And again, you have to look carefully at what's on the x-axis and what's on the y-axis. What's on the y-axis here is the intensity of sound that's needed to drive an auditory nerve fiber, a single auditory nerve fiber, to threshold. In other words, the intensity of sound that's needed to generate action potentials in that particular auditory nerve fiber. So for this, and what's shown on this axis is the frequency of the sound. Again, the ear is being presented with sine waves of sound. And so if you are able to record from this particular auditory nerve fiber, and this is done by simply teasing the auditory nerve apart in an anesthetized animal and trying to put hook electrodes under just one nerve fiber, or maybe suck just one nerve fiber into a micropipette. If you're able to record the action potentials in this nerve fiber, this particular nerve fiber, you'd find that it takes a very low frequency of 10 kilohertz sound to generate action potential. Sorry, a very low intensity of 10 kilohertz sound in order to generate action potentials. But it takes quite a high intensity of 1 kilohertz sound to generate action potentials. And so this cell is said to have its lowest threshold, that is its highest sensitivity, at 10 to 10 kilohertz sound. All right, so this is the sensitivity in decibels. Sorry, this is the intensity of the sound in decibels that's being presented. And minus 80 means that a very quiet sound of minus 80 dB will generate action potentials in this nerve fiber when the sound is at 10 kilohertz, but that it needs a much more intense sound of about minus 30 or minus 30 dB or so to generate action potentials when the sound is at a frequency of 1 kilohertz. So this cell is said to be tuned, or this nerve fiber is said to be tuned to high frequencies of sound. Anybody clear about that? Yes?
So all of these different little graphs are different parts of the same nerve?
So each of these represents a recording from an individual nerve fiber.
From a different nerve?
From a different nerve fiber, yes. Within the bundle of nerve fibers that constitutes the auditory nerve leaving the cochlea. That'll be a bit clearer in the next diagram, I hope. So you can see that some auditory nerve fibers are sensitive, most sensitive to high frequency sounds, some to middle frequency sounds, and some to low frequency sounds. So each axon is tuned, most sensitive to a particular range of frequencies of sound. So what does this mean in terms of the anatomy of where these these individual nerve axons are connected to? It turns out that, of course, nerve axons that are sensitive to the lowest frequencies, that are most sensitive to the lowest frequencies, like this one here, are receiving their input from hair cells in this part of the basilar membrane. And unfortunately, this diagram is 180 degrees reversed compared with this diagram, right? Here's the stapes and the oval window over here, and here's the oval window over here on that line. But remember that the basilar membrane, that is part of the basilar membrane that is furthest from the oval window, is going to vibrate most in response to low frequencies of sound. And therefore it's not surprising that a nerve fiber that is making synaptic contact with the hair cells in this part of the basilar membrane is most sensitive, this is its tuning curve here, to low frequencies of sound. Okay? Whereas a nerve fiber that makes contact and receives synaptic input from hair cells up here, close to the oval window, is one of the ones that's going to be most sensitive to the highest frequencies of sound. Okay, so The tuning results from the fact that each axon is connected to a hair cell at a different place along the basilar membrane of the cochlea. And so this tuning by position, whereby sound frequency, that is sound pitch, is coded in terms of the position of these nerve fibers when they leave the basilar membrane. This tuning by position is the only way, actually, that the brain can discriminate the frequency, that is the pitch, of high-frequency sounds. That's greater than about 3 kilohertz. So the only way our brain has of determining whether a sound has a frequency of 3 kilohertz or of 10 kilohertz or 20 kilohertz, these are relatively high-pitched sounds, is by figuring out where the axon that's showing that activity originated from along the basilar membrane. And of course, the only way the brain is going to be able to do that is if there is a tonotopic projection of these axons all the way up through the mid-lower brain, through the brainstem, through the midbrain, and into the cerebral cortex, so that axons which originate from neighboring parts of the cochlear membrane arrive at neighboring parts of the auditory cortex. We've talked about retinotopic maps, we've talked about somatotopic maps, and this is a tonotopic map, a map of sound frequencies. Okay? Not a map of position of the sound in space. Right? A retinotopic map preserves information in terms preserves information on the position of an object in visual space, where it is in visual space. A somatotopic map preserves information about the position of a stimulated part of our skin in relation to the other parts of our skin. All right? But a tonotopic map is different. It's preserving information about sound frequency, even though anatomically it's the same thing, right? The sensory surface is just projected point by point onto neurons of the cortex. Yeah? Okay, so above about 3 kilohertz, to discriminate pitch, we have to use this idea of a tonotopic mapping of the cochlea onto our auditory cortex, and we'll come back to that when we talk about the auditory cortex a little later. But what about at frequencies lower than 3 kilohertz? And why is it, what's magic about this 3 kilohertz number? So this shows you the electrical recordings from a hair cell. So this is 25 millivolts on this axis. So this is the membrane of the hair cell going up and down. And so this is what will be driving action potentials at the in the afferent nerve at the synapse between the hair cell and the afferent nerve, the one we've been talking about. And you can see that at frequencies below about 3,000 hertz, that's all these frequencies here, the membrane potential of the hair cell is actually following quite well the sound frequency. So as the individual pulses of pressure that comprise a 1,000 hertz sound hit the cochlea every millisecond, if it's 1,000 hertz, they each produce a small movement of the hair cell hairs, and that is reflected by a small depolarization. Okay, so this depolarization here is going up and down about 1,000 times per second. And you can see that here, this is 10 milliseconds, and you can see there's about 10 ups and downs over that time period. Yeah? So it's conceivable, and we'll see that it actually happens, that say for 1,000 hertz or 900 hertz or even a 500 hertz signal, an action potential could be fired in the afferent nerve, that is the nerve that synaptically connects to this hair cell. An action potential could be fired at the peak of every one of these little cycles of depolarization. So that the peak of these cycles of depolarization, a little burst of vesicles are released, a little burst of neurotransmitter arrives on the afferent nerve, and in consequence there's a burst of depolarization of the afferent nerve fiber, which is transmitting here the information from the hair cells, which are up here, down towards the brain along the auditory nerve. Okay, so that works up to about 3,000 hertz, and then above 3,000 hertz, the capacitance, the electrical capacitance of the hair cell membrane begins to slows down the way the voltage of the hair cell membrane follows, the membrane potential follows the movement of current through the mechanosensitive channels. And so above 3,000 hertz, what sound generates is just a smooth depolarization of the hair cell. Okay, and that's the reason it's a depolarization is because of the asymmetry of the voltage-current relationship that we looked at in the last lecture. So above about 3,000 hertz, we have to rely as mammals on the position of this particular hair cell along the cochlea. We can't get any information about the frequency of the sound from the vibrations of the membrane potential of the hair cell, right? Because it's just not going up and down over a large enough amplitude compared with the noise level for us to be able to say what's happening. Yeah. Yeah, it's a saturation point. It's not a saturation. It's more a filtering, so that the high frequency, the current in this case, the channels are still opening and closing at a very high frequency, and so the current is still entering and not entering and entering and not entering and entering and not entering at a high frequency. Yeah, but because of the membrane capacitance, the charge is not being placed on the membrane fast enough to change the membrane potential fast enough. Yeah? And so it's more like you can think of it as a high-frequency filtering mechanism that filters out the high frequency. Yeah?
So the difference, so we can hear the difference between like 300 and 1,000. We can't hear the difference between 3,000 and above?
Yes, we can. We can hear the difference between these by relying on the tonotopic mapping of the position of the stimulated hair cell in relation to the cochlea. Yes? Our brain figures out where which hair cells, which afferent nerves are active, and where they originated from on the cochlea. Okay, and the brain's able to do that because of this precise mapping, tonotopic mapping, of the arrangement of the cochlear afferents, right, as they project to the auditory cortex. Yeah, in the same way as we're able to predict where a flash of light is occurring, because the projection from the retina surface to the visual cortex is retinotopic, right? Neighboring parts of the retina project to neighboring parts of the visual cortex. So this putative little guy who lives in our head can determine where things are happening by determining which cortical neurons are active in relation to which others. Yeah? Yes? Okay. But, so, what I'm saying is that we can determine, obviously, we can determine the pitch of all of these sounds, but the strategy that we use for the high-frequency sounds is different from the strategy that we might use for the low-frequency sounds. For the high-frequency sounds, we can't rely on just counting when the action potentials occur. Yeah? We can only rely on the tonotopic mapping. And what this means is if you find an animal without a cochleus, and it's really only mammals that have cochleases, you have to find, well, fish have something like a cochleus, but if you find a mammal without this kind of basilar membrane arrangement, you have to consider that it might not be able to discriminate between one high-frequency sound and the next, unless it's using some other mechanical trick. separating out the frequencies. Yeah, Anthony, and then well, this one. No, Anthony, I think you were second, so we'll go here. That's you. Yeah.
Do you mean just like we can determine what the sound is, like looking at it experimentally, or saying that our brain processes both the frequency and the position for the lower sound?
So for the lower level sound, our brain is capable of determining the pitch of the sound, the frequency, either by counting when the action potentials occurred, right, which is equivalent to counting these individual depolarizations of the hair cell, in a way, yeah, or just the position on the cochlea. Whereas for the high frequency sound, we have to rely simply on position.
Do they know that it's the frequency?
Sorry, do they know which one?
For like lower level sounds, do they know that you can just like frequency in your sound rather than just position?
Yes, you can do that. And remember that there are many animals that can hear low frequency sounds that don't have cochlea, can discriminate low frequency sounds. They don't have cochlea. So they're doing it as well. Yeah, and there was, Anthony, you had a question. Same question. I mean, does it really happen both ways? They are redundant, yes. Yes. So this just shows you how the firing of the action potentials in a afferent nerve fiber in response to a low frequency sound. So this is the sound vibration pressure for a low frequency sound, just like this would be what this hair cell is following, say, at 300 hertz. And this is a histogram of when action potentials are likely to occur in the afferent nerve fiber. So sometimes action potentials may occur here, 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 and here, and here. And then you present the sound again, and action potentials might occur here, 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 and here. And you just work out a histogram of when is it most likely that the action potentials will occur after the sound. And you can see that it's most likely that the action potentials will occur at exactly the same frequency, with exactly the same frequency as the sound frequency here. In other words, there's a high probability that an action potential will be fired here, and 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 here. Okay? And so this shows you how the information about sound frequency that's given to the brain for low frequency stimuli is quite precise. Yeah? Although it might take some presentations of the stimulus to be able to figure out the precise frequency. And so this is the kind of information that an animal that doesn't have a cochlea might be using to determine sound frequency. Okay, so what happens to the information after it leaves the cochleus along the auditory nerve? So here's the auditory nerve here, and it terminates in the medulla here, in the brainstem, in a region known as the cochlear nuclei, and a group of nuclei known as the cochlear nuclei. And you can see that the auditory nerve fibers branch and innervate several sort of subnuclei within the cochlear nucleus here. So this is coming from this ear, and obviously there'd be an equivalent projection from that ear. Now, still in the brainstem really, or in the upper brainstem or the lower midbrain in the pons, there is a structure known as the superior olive. Again, this is a little nucleus of cell bodies, and this is where cells are located that are receiving information from the cochlear nuclei cells. So these are the sort of actually the second order afferent fibers, because the hair cells in the way are the first sensory neurons. So these are the sort of second order afferent fibers. Then you've got your third order afferent fibers here, and then the fourth order ones have their cell bodies in the superior olive, and they send axons up to the midbrain and the pons here. You can see a branch there to nucleus of lateral meniscus in the pons. And they also send branches up to the inferior colliculus here. in the midbrain. And then finally, the neurons in the inferior colliculus, which receive information from the olive, send axons up to the thalamus. So we finally arrive up here at the medial geniculate complex of the thalamus after going through 1, 2, 3, 4 layers of neurons. Yes? Yeah, so that's right. The crossing, the main crossing, or the one we're going to learn about, is this one here at the level of the pons. And we'll talk about that because it's very important for sound localization. But I believe that there are crossing points elsewhere. Just because these tracks are shown just up one side doesn't mean that they necessarily aren't crossings there. Yeah.
Can you say again where the first, second, third, fourth?
Yeah. So I mean, this counting is a bit. You don't want to sort of get too sort of obsessed by the counting. But the first neuron to consider is probably the hair cell. Right, and then this hair cell connects to the auditory nerve afferent fiber. So that would be either the second or the first, depending on how you put it. What should we call it? Should we call it the second? Okay, we'll call that the second. And then the third one is in the cochlear nucleus. Then the fourth one is in the superior olive. And then the fifth, and so it takes 4 to get up to the inferior colliculus. And then by this counting, it's 5 to get to the thalamus, because we're counting the sensory neuron either side as the first. Okay? Yeah? Right, so that's too many to remember. So what I want you to understand from this is it takes a bunch of connections to get to the thalamus. First point, right? How many connections did it take to get from the retina to the thalamus? How many from the retina to the thalamus? The retina, not the photoreceptors. 1, right? Because the retinal ganglion cells project to the lateral geniculate nucleus. Done. Yeah? So to get from the photoreceptors, there's the photoreceptor, the bipolar cell, the lateral geniculate nucleus cell, the retinal ganglion cell. So that's 3 to get from the photoreceptors. Yeah? What about the somatosensory system? How many to get from your the tip of your finger to the thalamus, say? It's 1 is the sensory neuron, 2 is the one in the cuneate nucleus, and then that goes to the thalamus. So it's 2. Okay, and it's the same but with a different crossover point for the... So the numbers of neurons, the number of way stations, as it were, right, between the sensory organ and the thalamus and the cortex are much larger for audition than for somatosensation or vision. Okay, there's many more relay stations on the way up to the thalamus and the cortex. And when did the crossover, or when did information combine from the 2 eyes? Where's that occur? Where is there convergence of activity from the 2 eyes? Yeah, actually it's the upper and lower layers of cells in the visual cortex, not even layer 4 of the visual cortex. Yeah? All right, so way up there, right? So the first crossover, the first combination of information from the 2 eyes occurs all the way up there in the visual system. Yeah? So again, that's a big difference, because the first combination of information from the 2 ears is going to happen here in the superior olive. Okay, so there are some differences, and I hope by the end of the next couple of slides you'll realize why there are differences. And so what I'm going to do is going to tell you a story about the superior olive. And this story features horror redundancy. So there are 2 ways of doing the same thing in this story. And I don't know which one is superior, but I suspect we use them both. And different animals use the 2 methods for different extents. So we're talking about the processing of sound information at the lower brain centers, in particular in the mid-pons here. And we're talking about the role of the medial and lateral superior olives, that is the MSO and the LSO, in integrating information from the two ears and localizing the source of a sound. Because just as we need to use two eyes to localize the distance of an image from us, of an object visually from us, we obviously, we need to use two ears to localize a sound in space, usually. And there are two ways of doing this. One is an extremely smart way, which is the way that's calculated in the MSO, in the medial superior olive. And it uses the interoral arrival time difference, which is a long way of saying that the fact that the sound arrives at this ear, if it's coming from over there, the sound arrives at this ear shortly before it arrives at that ear. Yeah? Yes? Okay. And it uses that time difference to calculate the direction from which sound is arriving. Okay, so obviously the brain can calculate whether it's arriving at this ear first or that ear first, then it can determine roughly where the sound is coming from. Yeah? Well, yeah, so again, there's an analogy. She's asking what happens if you're partially deaf in one ear. How can you do this trick of localizing sound? And as with vision, there's a way that people get around that. If you don't have binocular vision, if you only have one eye, for instance, or there's some damage of the optic chiasm or something, then if you don't have binocular vision, then you can figure out how far an object is away by moving your head and seeing how much the object moves in your visual field compared with the background. It's called parallax. Yeah? And basically, the further the object is away, the less the object moves with respect to the background as you move your head. Right? Makes sense? Yeah? Because we're all close up, it's going to move a long way. Okay? So we use parallax to determine, we all use parallax to determine distance, as well as binocular vision. And in fact, binocular vision really only works for a limited range of distances, because remember, the binocular vision depends on the brain calculating where the image arrives in the two retinas. And it can only do that for relatively short distances apart for the two images. Discrepancies, relatively short discrepancies. And so for vision, we have a fairly limited range where we do true binocular vision, and everything outside of that, beyond that, we tend to use parallax. And so with ears, it's the same. You can block the sound coming from one ear, or with your head, and use your pinner here to move your head and try and figure out how the intensity of the sound changes as you move your head, and figure out where it's coming from. Yeah? Okay, obviously if it's coming from behind you, it's going to be softer than when it's coming from the head. But you'll make mistakes. Okay? And then the lateral superior olive uses interoral intensity differences between, obviously, sound that comes from that side is going to be louder than sound, is likely to be louder from sound that's coming from this side. Okay? So let's look at the mechanics of this process. And this diagram here, it's not strictly accurate because it seems to suggest that there is a single axon, a single neuronal axon, that leads from the cochlea all the way to the medial superior olive, which is diagrammed in this structure here. And we know that that's actually not the case. There's a couple of neuronal axons, a couple of neurons involved in this transfer of information. Okay, so the diagram simplifies that. That's why it says cochlea and cochlear nucleus. It really means cochlea via the cochlear nucleus. And if we were going to be correct, we'd have another cell body synapse in here. Right? But it simplifies things just to have the sound being transmitted along some axonal pathway to the medial superior olive. And it's transmitted from one ear here, and then, as we can see here, it's transmitted to both the ipsilateral superior olive and the contralateral superior olive. So any one of these superior olives will be receiving information from this ear and from over here we'll be receiving information from that ear. And so this shows the projection from one ear over here and the other ear over here. So within the MSO, there are a series of neuronal cell bodies, and which are labeled A, B, C, and D here. And these neuronal cell bodies will be sending axons further up into the midbrain and towards the inferior colliculus. All right? So they're not shown there either. But the idea is that sound that comes from one side of the body will stimulate certain of these neurons more than the other neurons. And I'll show you how that happens in a moment. So you'll notice that each of these neurons has two axons coming into it. One is a branch from the axons that are coming from the right ear, and the other is a branch from the axons that are coming from the left ear. Yeah? And you'll notice in this diagram that there is physically a shorter distance for the right ear to reach neuron E via this axonal path than for the left ear to reach neuron E from the left ear over here. Okay, just there's a longer pathway here and a shorter pathway here to neuron E. Okay, so let's imagine that a sound originates on the left-hand side of the body over here. So the sound pressure differences are going to reach the left ear first. And the sound pressure differences are going to reach the right ear a few microseconds later. Then in terms of what neuron E receives, neuron E will in fact receive action potentials at the synapses here simultaneously from both ears. Because although the sound reaches the left ear first, it's going to take a longer time for the action potential to travel along that slightly longer pathway to neuron E than it is going to take the action potential to arrive from the right ear. Okay, and so stimulation of the left ear will generate the arrival of simultaneous action potentials at neuron E. And as we know, simultaneous action potentials create simultaneous end plate potentials or excitatory postsynaptic potentials, and they will precisely sum, because they're occurring at the same time, to create a larger combined EPSP that will drive neuron E to threshold and generate action potentials in neuron E. Right? Whereas the action potentials will arrive at neuron A in an asynchronous manner. Right? They'll come first here, and then this one, which not only starts later, but has a longer pathway, will arrive a little later. Right? And these neurons here are specialized so that the amplitude of the EPSP and the synchrony of these two EPSPs is enormously important in determining whether they reach threshold or not. Right? The EPSPs don't last for very long. It's a very brief event in these neurons. So they have to be synchronous to generate a combined EPSP, a summed EPSP, a temporally summed EPSP that reaches threshold. Okay, and obviously you can see that this one in the middle here, this guy here is going to be most sensitive to sound that is coming from the middle of the body, and this one here is going to be most sensitive to sound that's coming from over there, and you can figure that out for yourselves in terms of the pathways. Okay, so what we have here is a map of auditory space which is coded in terms of the position of these neurons within the MSO. And you can see how if the axons from these neurons project in an ordered manner, so that neighboring axons project to neighboring parts of the inferior colliculus, say, that map is going to be projected up into the higher brain centers. And so just as there's a tonotopic map displayed amongst the higher brain cells, there's also a auditory space map there as well. Okay, but that's not the only way in which the midbrain, or the pons, calculates the intensity, the positioning of a sound. The other way is a bit cruder and in some ways easier to understand. If you have a speaker that's placed against this ear, or a sound that's coming from this side, it's obviously going to be louder in this ear than it is in the other ear. And so this ear is going to project, this ear here is going to project directly to this part of the lateral superior olive, and you can see it makes a direct excitatory connection to cells in the LSO that send action potentials up to the inferior colliculus and the higher brain centers. So there is direct excitation from activation of this ear, and then there's a branch here of the same axons that goes across the midline, and we talked about these branches that go across the midline. There's a branch here that goes across the midline, and via an inhibitory interneuron here, it will inhibit action potentials produced by the contralateral LSO neuron. So that sound arriving at this side will excite on this side and inhibit on that side. And so there'll be a large net difference in activity if the sound is coming from this side between what's being passed up here to the high centers and what's being passed up here to the high centers on the contralateral side. Yes?
These methods could distinguish a sound on the right or left or right, but I don't understand how they can distinguish a sound that's on the...
No, so they can't do that. And to do that, I think you have to turn your head towards that sound, or you have to rely on just the intensity. You have to use the flaps on your ears to rely on the fact intensity differences between what's coming ahead and what's coming behind, which are because of the shielding that your pinna provide, right?
I couldn't tell that you're talking when you want to be...
You can tell, if you know I'm talking, you can probably tell if I'm talking in front of you compared with behind you, because I sound quieter behind you than I do in front. Yeah, and you have a sort of mental image of what my voice is like. But I'm not sure. I mean, I might be wrong, but that is certainly a tricky question. You know, and then there's the question of what about up here versus down there, and that's why one of the reasons for discriminating those differences is why we and other animals have such complicated structures, baffles as it were, around our ears, so that we can use them like, you know, antennae or radar dishes, and by moving our heads, localize the sound in space up there. Because again, these methods don't work from here versus here. Yeah. So, but then, you know, we move our eyes all the time as well. So we do the same thing with our eyes. Okay, so we talked about, so if you map just as similar, if the relative loudness is high on the left, if the left is more intense than the right, so the sound is on the left, then there's a high LSO output and an inhibited right LSO output on the left versus the right. And then over here, if the sound is on the right, you get a high output from the right LSO cells and a low output from the left LSO cells. And so it's like a flip-flop. Yeah? I don't know the answer to that. You know, there are certainly auditory neuroscientists here, perhaps, who could venture that one. But firstly, is never underestimate evolution. That one may have evolved first, and we still hang on to it because it's useful. The second is to say that this business of let me do some explanation here. So the difference between the arrival of sound here and here is a matter of microseconds, the timing. Right? Now you think about how long an EPSP lasts. It lasts a few milliseconds. And we're asking a neuron to discriminate between the arrival time of two EPSPs, right, that are occurring within 10 microseconds of one another, right, compared with occurring within 50 microseconds of one another, depending on where it is, right? So this is, I don't think, an easy task. I think it's a noisy task. In other words, we can't be relied upon to make the judgment 100% every time. And that may be why we correlate two different methods, because that's a very powerful way, always, of reducing noise in a data set, is by doing your calculations in two different ways and taking the solution, accepting the solution that comes up using both methods. Okay, so what about we're going to talk very briefly about the inferior colliculus. Here, in the inferior colliculus, neurons with cell bodies in the inferior colliculus are arrayed in such a way as to create an auditory space map. We talked about that because this is where the MSO cells and the LSO cells are projecting to. Neighboring neurons are preferentially sensitive to sound coming from neighboring directions. That's what an auditory space map means. And we've talked about how it's calculated based on the output of the LSO and MSO neurons, which converge on neurons in the inferior colliculus. And this map is highly developed in nocturnal predators, like barn owls, for instance. Very highly developed. They have beautifully arranged inferior colliculus auditory maps. And this was all worked out by Catherine Carr, who works in our department many years ago, but she's still living off the proceeds and continuing to discover things about, beautiful things about animal auditory systems and comparative biology auditory systems. And it's a wonderful example of how by studying an animal that takes a particular behavior to an extreme, like a nocturnal predator. I mean, a barn owl can find a mouse in darkness, just from the way it's rustling its way through the undergrowth, and make a perfect strike. Yes? Yeah. Yeah, barn owls have these, well, they have these complex sort of arrangement of feathers that create these sort of bowl-like surfaces around their ears. And one ear is slightly displaced relative to the other, so that they can figure it. There's a slight difference in the timing from sounds that come from above and below as well. Yeah?
So how do bats compare?
Yeah, I think we'll talk about bats in a moment, perhaps, because that's to do with the next layer of cells. But what bats have are they have firstly what they do is they scream a sound at a particular frequency at an object, and then their auditory system is highly specialized to pick up that sound and calculate the timing difference between when the sound was emitted and when the sound arrived back. And so it's that timing difference that they're looking at. And again, that's calculated, I think it's calculated in inferior colliculus, or at that level, roughly. But you'll find, again, you'll find whole areas of the brain there that are specialized for that task. Okay. So what about the thalamus? You'll find neurons here that involved in the detection of specific complex sound patterns. So yeah, so echolocating neurons in the bat, thalamus, it's in the thalamus, not in the are sensitive to particular delays between the time of emission of sound and the time at which the echo is received. What about the auditory cortex? And the auditory cortex is a somewhat more mysterious structure and much less well investigated than the visual cortex. So the auditory cortex is where you find the most prominent examples of this tonotopic map, an array of neurons according to their characteristic best sound frequencies that they respond to. And just a note that this is in effect a topographic map of the basilar membrane. And in addition, in the cortical neurons, you find in the cortex, you find neurons that can be binaurally stimulated. That is, they can respond to sound from both ears. There are strips of neurons excited by one ear, but not the other, which alternate with strips excited by both ears. Similar in a way to those ocular dominance columns in the visual cortex as well. And cortical neurons are also particularly sensitive to certain sound combinations. So certain combinations of frequencies of sound will best stimulate them. And so here we're getting into the realm of neurons that are sensitive to particular classes of vocalizations and frequencies that an animal might actually use or respond to. And that's all I'm going to say. The auditory cortex is in this part of the temporal lobe. Here is the secondary auditory cortex here, and the primary auditory cortex is just sort of tucked underneath this fold here, on the other side here. If you sort of take a shovel and pick open that fold, dig it into the fold and kind of pry the corner of the fold up, the flap of the fold up, you can see one of these areas where if you recorded with electrodes and measured the best frequencies of sound that these neurons respond to, you'd find this sort of tonotopic map going from 500 hertz to 16,000 hertz in this case. Tonotopic organization of characteristic frequency of auditory cortex neurons. Okay, so that's all I'm going to say about audition. It's not as well understood as vision in terms of what features the brain uses to characterize a sound by. And for that reason, it's a really fertile area still for people to get involved in. I think the main questions in vision are quite complex. They involve the binding problem and very complex quite complex stimuli, face recognition, for instance. In audition, there are still many problems at the sort of basic level to understand and to characterize. But I'd like to pose the question now is, why is there so much information processing going on at the lower levels of the auditory system compared with the lower levels of the visual system? Why is it that the sound is combined from both ears at a very low level? And why is it that at the lowest levels here, there's sort of calculations being performed to calculate a map of auditory space? Why does all that happen at such a low level? Yeah?
I'm guessing it's because, like what you mentioned earlier, with each time you convert it, creates potential doesn't that decrease the specificity that your sound is coming in at? So it's like at every level, you sort of decrease the specificity, so you want it to create it as early as possible.
So you're saying that at every level you decrease the specificity of the output, as it were, of when the action potentials occur, and so you want to do that at a lower level. But why do you want to do it at a lower level for the sound, for the auditory system? Why is that? Why is there a need or drive for that? What is so special about these calculations here? It's more primitive. That's a dangerous word to use. No, I don't think that's. I mean, that might be part of it, but actually, if I was going to look for a primitive sensory system, I'd choose the smell system, and I don't think the visual system is that far behind. Yeah.
Hearing is highly dependent on time and distances of neural tracts.
Yes.
So maybe if you had higher processing and cross-correlation going on farther up in the brain, there'd be more chances to mess things up if one neuron is a tiny bit longer in some section.
Yeah, that's maybe what you were thinking of, but you didn't mention that the specificity that you were talking about was time. Yeah, that these calculations and presentations of information, be it the timing of when neurons, be it the timing of when signals arrive in the MSO, or be it the timing of these action potential bursts of when action potentials might occur during a stimulus. It's all about timing. And the timing is very tight. It's on a sort of millisecond resolution, a microsecond resolution in some cases. And just inevitably, as an action potential moves down an axon, or as it crosses a synapse, there's going to be some jitter in the time that it takes to do that, from one trial to the next, from one stimulus to the next, or from one peak of vibration to the next. And so if you can do all your calculations early on, you can minimize the impact of that noise source. Yes?
So, as in, like, if we do all the calculations early on compared to do it later on, if we did all the calculations later on, and something messed up in the beginning, and it's like not as bad as an effect, because if you're calculating this early, and if there's a small mistake later, then at least the whole thing's still kind of cohesive.
Well, I think it's easiest. Yeah, it's easiest to see if you think about the MSO system here, right? So once the auditory map has been coded as a position, timing is no longer of importance. Yeah? But while the auditory space map is essentially coded as a timing difference between how long it takes an action potential to go along here and how long it takes an action potential going on there, I hope you can see that keeping both of these pathways as short as possible will increase the accuracy of that comparison. Yeah? Yes?
Do you know if there's a certain level of the transduction pathway where most auditory complications lie, or is it kind of just all over?
I don't know. I think in both the auditory and the visual system, the complications that we know most about, because they're the ones that doctors now have a chance of handling, are all in the sensory organ themselves. And that concerns the implants that are made in the case of patients with auditory deficiencies who are deaf or in terms now of even retinal implants as well. So that's what, by and large, what the literature is about. But you probably have to ask that question as a physician in terms of what comes into their office. But I bet the answer would be the same. Because the brain has, as you've seen, there are redundant pathways in the brain. And the brain has a way of working around issues. And actually of concealing them, so you don't know that there's a problem. Whereas if the problem is low down, as somebody pointed out, then it's a major issue because it can't be circumvented. Yeah, it's in there. So understanding this can also, lead to some understanding of the problems involved with cochlear implants, because in order to sort of revive how the perception of sound, all you have to do is connect some kind of a stimulator, electrical stimulator, to these nerves. And then you can generate rhythms of activity in those nerve fibers that will be perceived as a form of sound. But if you want to accurately restore, say, a musician's sense of hearing, pitch, then you have to make a cochlear implant that is going to precisely stimulate different areas of the cochlear nerve, different cochlear nerves, depending upon the frequencies that the amplifier that it's connected to a microphone picks up. And the sort of state of the art of cochlear implants is trying to improve the sort of artificial representation of the basilar membrane and restore pitch sensation. Because my understanding is that retinal implants don't convey a sense of pitch, at least anywhere near what a musician would need. Okay. This is the point where he realizes he left his pen behind. Oh, God. We're still going to get out of the bloody sensory systems. Damn. Doesn't matter. I like sensory systems. My show. There we go. Okay, your nose. All right, very fond of noses. So these are your nostrils, right? These are your teeth. I guess here are your lips. Just to orient you. And this is the big cavity that exists behind your nose, right? So this is the part that fills up with snot when you have a cold. It comes out here, right? Drips out there. And up here is the olfactory epithelium. So up here is the epithelial tissue that contains the olfactory receptor neurons, that actually detect smell. And we have really just one olfactory epithelium here and one olfactory bulb. So the neurons, the sensory neurons in the olfactory epithelium project and make synaptic contact with, project to and make synaptic contact with second order neurons in the olfactory bulb. And then they send axons to other parts of the brain. And these little black things here are the sensory dendrites of the receptor neurons. So these are where the molecules are that perform olfactory signal transduction. And this space here is, as you well know, is sort of filled with a mixture of fluid and proteins and mucus, and so it's not an air-filled space quite, as it seems to be represented here. Now, the olfactory receptor axons pass directly into the brain. There's no involvement of, this olfactory bulb here is actually an extension of the brain. There's no pathway through the thalamus here. And to some extent, this is a more sort of ancient method, a more direct pathway, as we'll see, to certain brain centers, also ancient brain centers. One thing to note here is that this cribriform plate, this bone here, is a bone through which the axons of the receptor neurons, these guys here, have to pass to get to the olfactory bulb and make their synaptic connections. And so if you're punched in the nose, these olfactory axons get broken, and so anosmia, the lack of ability to smell, is a common consequence of being punched in the nose or being a prize fighter or any sort of activity, falling flat on your face or any sort of activity like that. Okay, so anosmia in that sense can last for several weeks while these receptors grow new axons. And the other interesting thing about these olfactory receptor neurons is that they are continuously renewed. They're one of the few neurons in the central nervous system that can be replenished from stem cells, which are located in the olfactory epithelium. Okay, the photoreceptor cells can't be replaced once they die. Hair cells in your ear can't be replaced once you die. That's why you shouldn't go to very, very loud rock concerts without having some kind of ear protection. Right? So the amazing thing about olfaction is the variety of chemical compounds that we can sense and distinguish one from another. And so here's just a smattering of different kinds. This one here is, this compound here smells like bell pepper, violet, cucumber, putrid, urinous, moldy, musky, ethereal, alcoholic. Okay, so each of these compounds, and if you can find rhyme or reason that connects the chemical structures to our perception of the smell, then good luck to you. Obviously, small changes in chemical structure will generate a smell that's very similar to another molecule with a very similar structure. But on the whole, they're all got hydrophobic bits. They've all got some rings in them. They've all got some pi bonds and oxygens. They all look partly polar and partly non-polar. There isn't a sort of a rhyme or reason here that's easy to see. And there are a tremendous variety of these compounds. And so, unlike rhodopsin, right, what rhodopsin has to detect is really the presence of all-trans retinol, right, which is already bound to it. Yeah, light hits retinol and isomerizes it. So all that rhodopsin has to receive and detect is all-trans retinol. All a neurotransmitter receptor has to detect really is the neurotransmitter and anything that's very similar to it. But the olfactory system has this enormous problem that it has to detect all these odors. So it should come to no surprise to you that genes for olfactory receptor proteins constitute a substantial fraction of our genome and of other animals' genomes. So I think in the human genome, I think it's human, there are like 500 genes for olfactory receptor proteins, all of which are variants on your basic G protein coupled receptor. Okay, so they're all G protein coupled receptors, they all look alike to some extent, but they each are different in terms of their specificity of binding to different combinations of odors or different odorants. So these are called odorants. Right, so here's our a blow-up of the olfactory epithelium. The odorants arrive here, that little green molecules here, and then there's this layer of mucus which sits on the top of the nasal cavity. The dendrites of the olfactory neurons are embedded in this mucus, and so for many olfactants there are actually proteins which bind to the olfactants and carry the olfactants through that mucus layer and protect them from enzymes and other things within that mucus layer and deliver the odorants to the dendrites of the olfactory cilia. But that's not a necessary part of signal transduction. So what's going to happen here is that the dendrites are going to detect the odorants. The dendrites are then going to produce a generator potential or a graded potential change across their plasma membrane, right, in much the same way as we've seen in all the other sensory cells that we've met. And then that generator potential is going to spread passively into the cell body. There will be voltage-gated sodium and potassium channels that generate an action potential. So the graded generator potential will be converted to an action potential, and then the action potentials will travel up through the axons into the olfactory bulb. And we'll say now I'll tell you now that the generator potential, the graded potential that's graded with the amount of olfactant that's present, is a depolarizing event. So we've got none of that weird hyperpolarizing stuff that happens in rods. This is a good old-fashioned depolarizing generator potential that will therefore directly produce action potentials that will travel up through the axons into the brain. So to demonstrate that it's the dendrites that initiate the signal and that contain the olfactory receptor proteins, there's a simple experiment you can do. You can use a whole cell patch clamp to record the current that is flowing into this olfactory receptor neuron, here and here, and then you can spritz, you can spray odorant onto the dendrites or onto the cell body. And obviously to do this you have to have dissociated the olfactory receptor neurons from the epithelium and put them in a dish, and then you sneak up on them with your patch clamp electrodes, whole cell patch clamp. And so when you put the odorant onto the dendrites, you see a large inward current flowing into the olfactory receptor through the whole cell patch clamp. And that's shown here. It's a negative going inward current, negative going trace. If you put the odorant onto the cell body, almost nothing happens at all, and it also happens with quite a long delay, because what we're probably looking at here are odorant molecules that are managing to diffuse to the dendrites, as many as get there. Okay, so this proves that it's the cilia, right? And these dendrites turn out to be cilia, just as the outer segment of the rod is a modified cilium. So the outer segment of, so the dendrites of these olfactory neurons are also cilia, except that there are several of them, not just one as in the rod. And they actually do have cilia, I think, that run, actually have microtubers that run their whole length. Okay, so question. Yes?
So are the generator currents in or out?
The generator, this shows a downward trace, and so the generator currents are flowing into the cilia.
Why
because inward current is shown as a downward trace. Oh yeah, right. I've got, yeah. Okay. Okay. So what's the time do we have? Do we have 1:42? Another 3 minutes. Oh. 1:43. Another 2 minutes. Okay. So these inward generated currents are seen under voltage clamp here, or patch clamp, wholesale patch clamp, will then produce, under natural circumstances, they would produce a depolarization that is graded with odorant concentration, and then that depolarization would generate action potentials. So these are extracellular recordings that are made from the olfactory neuron axons up here, and you can see that this odorant here, presentation of odorant here, produced a little burst of action potentials, and here there are big bursts, and here's an even bigger burst of action potentials. So this simply shows you that the frequency of action potentials increases with increasing odorant concentration. And of course, in order to do this, you have to first figure out which of these many odorants your particular receptor here is actually sensitive to. And just to leave you with, we'll be talking a little bit more about this. We've got another few slides to get through on the smell system. But there is somebody in our department, the person who knows most about this is Ricardo Araneda. Oh, so if you want to endear yourself to Ricardo, and then do ask him about this and tell him I'm making an absolute matter of explaining it. Please explain. Yes.